Nisin is a bacteriocin, which is secreted by Lactococcus lactis, a gram-positive bacterium. It is classified as generally regarded by as safe as the FDA. It is active against gram-positive bacteria and has been used as a preservative in the food industry. However, it leaks out in fatty environments. Recently, it has attracted interest from the cosmetic industry as a potential natural preservative agent for skin formulations. Chitosan is a biopolymer that is used as a drug delivery agent. It is an inexpensive substance that is obtained by deacetylation of chitin, which is a natural substance found in marine animals and fungi. It shows antimicrobial properties against both gram-positive and gram-negative bacteria and has modest antioxidant and antifungal properties. Another feature of chitosan is that it has the ability to cross-link with different agents such as sodium tripolyphosphate and make nanoparticles, which can be used as delivery agents useful in the drug and cosmetic industries. Tannic acid is a powerful antioxidant that protects against free radical damage caused by exposure to ultraviolet light and thereby may reduce the risk of skin cancer and premature aging. It contains phenyl groups that form hydrogen bonds with the carbonyl groups in proteins, and due to this, we expected that tannic acid may modulate the release of nisin from the chitosan nanoparticles and enhance the antioxidant properties of the particles. As you can see here, we made nanoparticles with chitosan encapsulating nisin inside the chitosan shell using different ratios of tannic acid to nisin and also different concentrations of nisin. After we synthesize the particles, we characterize them using encapsulation efficiency, EE, and loading capacity, LC. The formulas used to calculate them are shown, and we also calculated the ratio of residual amino groups, which predict the positive charges in the nanoparticles and reflect the cross-linking efficiency. As you can see, both EE and LC increase with increased nisin concentration. They decrease in size with increased nisin encapsulation. Next, we determine the size and zeta potential of the nanoparticles, and these results show additional cross-linking and the decrease in size with increased nisin concentration. They indicate that tripolyphosphate and chitosan interaction form nanoparticles, and the encapsulation of the protein promotes aggregation. We also took FTIR spectra, which are shown on the slide. Spectrum A shows the variations in the spectra with differences in nisin concentration, while spectrum B shows the difference is in the spectrum, and this is due to the different concentrations of tannic acid inside the nanoparticles. We wanted to study the kinetics of the release of nisin, and as you can see here, these are the results from the release. In spectrum A, you see a initial burst at the beginning of the graph of nisin, and then after that we have a sustained release. Spectrum B also shows the sustained release, and you see in figure B, we have the nisin release from composites with the same amount of nisin without tannic acid or with different nisin tannic acid ratios. We studied the release in different lipophilic environments. First, we studied the oil and water emulsion, then the water and oil emulsion, then the synthetic human sebum. The least lipophilic was the oil and water, and then the most lipophilic was the synthetic human sebum. This shows the mass release over in the oil and water, and we see the highest release over time. In the nisin chitosan 0.5, we have a sustained release over time, and this was taken around 7 days, so Periodically, the release was checked, and as we see, higher tannic acid content in the composites, the amount of nisin released increased with increased lipid concentration in the emulsions, so it was greatest inside the human sebum. We also performed an antioxidant assay to determine the antioxidant properties of the nanoparticles. And as we see here, the antioxidant concentration, equivalent Trolox concentration, Trolox was our standard, it increases with increased nisin, and it shows that it has antioxidant properties. Also, with greater ratio of tannic acid, that also shows higher antioxidant properties. After that, we wanted to determine the SPF of the particles. The SPF stands for sun protection factor, and it represents a sunscreen's ability to protect against a specific portion of ultraviolet light. We tested two different samples, the 10:1 ratio and the 5:1 ratio of tannic acid. And we showed that the 10:1 ratio, the SPF was 3.5, which does not provide any UV protection. However, 5:1 ratio sample, the SPF was calculated to be 5.6, and this provides modest UV protection according to the European Commission recommendation. And interestingly enough, the 10:1 ratio with the lower SPF had a lower antioxidant property, but the higher ratio, 5:1, had the greater SPF and also showed higher antioxidant activity, showing a link between antioxidant activity and SPF. To conclude the research, the nanoparticles were characterized using EE, LC by size, and all the particles showed positive values of the zeta potential. These results were confirmed by colloidal titration, and the RRAG values calculated can also be interpreted as a proof of cross-linking. The positive charges present make our nanoparticles promising as antimicrobial agents. We took FTIR spectra, which proved cross-linking, interaction with tripolyphosphate, and increase in hydrogen bonding network. The encapsulation of nisin in the nisin chitosan matrices seems to be beneficial for nisin used as a preservative, as it shows a gradual yet sustained release. The presence of tannic acid has the opposite effect, which could be due to complexing the peptide as it speeds up the release from nanochitosan. All of the nanoparticles showed antioxidant activity, and nisin encapsulation increased, while tannic acid afforded a greater enhancement as expected. Higher concentrations of tannic acid provide modest UV protection, whereas lower concentrations do not provide any UV protection. The nanocomposite materials were prepared, which encapsulate nisin in a hydrophilic matrix, thereby protecting it from proteolysis and improving the kinetics of its release in hydrophobic environments. This, together with their antioxidant activity, recommends them for applications as natural preservatives and food in food and both skincare products. That is the end of our presentation today. Thank you all for listening.